Hi everyone, and welcome to our first genetics lab. Now, I know you would probably much rather be in the lab room itself with some hands-on activities, but I will try to make this virtual online experience as painless and as valuable as possible. If at any point for any of the slides or for any of the lessons we do, you have any questions or concerns, please contact me in the Remind app at any time, day or night. Now, with this first lab, what we're going to cover is some general lab safety, and then we'll do a brief introduction to genetics lab and a little bit of Mendelian genetics. So the first thing with lab safety that I want to point out is that when we have you go into the genetics lab classroom, it is considered a BSL-1 lab, which means it's a biosafety level 1. Now, when you look at this slide, you'll notice that there are 4 different biosafety levels, okay? The higher the number, for instance, a level 4, the more hazardous or dangerous the samples that you would be working with are. So thankfully, in a genetics lab, in a classroom, in a university, we're usually BSL-1, and then if you take a course in the university, such as microbiology, then we up it to BSL-2. But you'll never in the classroom work beyond that. You won't go to BSL-3 or 4. Now, the way we categorize these, when I say that you would be working this semester in a BSL-1 lab, that basically means that any of the samples you would be working with are not really hazardous to your health, especially if you are healthy, what we call immunocompetent, meaning you have a strong, normal working immune system. The samples that you work with wouldn't infect you. So for instance, in the genetics lab, in a normal semester where we go into the classroom, we first have you working with Drosophila, which are the little fruit flies, and those don't make you sick. Or we have you working with what we call a non-pathogenic strain of E. coli. So the later part of the semester where you'd be putting DNA into bacteria, you'd be working with live bacteria, but they're a strain that wouldn't make you sick if you are a, normally healthy, strong person. Whereas in BSL-2, you start working with some of the microbes where even if you are normally healthy, you consider yourself immunocompetent, if you don't handle those BSL-2 microbes carefully, they end up posing a moderate risk to you and you could get sick. But thankfully, they're all strains where we have, therapeutic treatments available. And so you'd hopefully be okay in the end. Fingers crossed when I say hopefully. And then at levels BSL-3 and 4, that's when you work with the really nasty stuff, the stuff where people working in BSL-3 or 4 labs, they have to be very careful, follow very strict protocols, because those microbes that they're handling, even if you're someone who's immunocompetent, normally very healthy, fit, strong, and you would assume that you're safe to be in a lab, if you're in a BSL-3 or 4 lab, the microbes you're handling can make you seriously ill. And in the case of BSL-4, they're microbes that they're handling in those labs that don't have proper, vaccines yet. They don't have proper treatment yet. So they're very dangerous labs, and you won't encounter these unless you're working in the highest levels of, let's say, government and healthcare facilities, the real, specialized buildings or facilities. Now, I want you to know examples of what you would find in each level. So I want you to circle, star, highlight BSL-1. Make sure that you know that at this level, this is your genetics lab classroom, and you would be handling fruit flies, which you'll see in later slides we call Drosophila. So you'd be handling fruit flies or non-pathogenic E. coli. Then know that you would not be handling, for instance, tuberculosis, Ebola, smallpox. Those are all much higher safety levels, okay? So make sure in your notes you know what would you be handling at BSL level 1 and what would you not be handling, okay? So you would be handling fruit flies or E. coli. You would not be handling TB, Ebola, smallpox, even COVID. So right now, very popular, you would not be handling COVID in a BSL-1 lab, okay? You wouldn't even be handling Staph aureus. Those are all higher levels, okay? If you have any questions or concerns about this slide, please just send me a message in the Remind app, and I'll try to make it a little clearer. Now, when you are working in any lab, even if it's BSL-1 and you don't consider yourself too at risk with the microbes or the organisms you're handling, you have to keep in mind that you're also handling a lot of different chemicals and reagents that could be problematic for you. So no matter what level biosafety you're working, you always, always, always want to be wearing PPE during experiments. Now, thanks to the COVID pandemic, most people now know what PPE stands for. So that stands for personal protective equipment. And what are the two main examples of that in a classroom lab? The two main examples of PPE are a lab coat and gloves. Okay, so in the real world during pandemic times, when you think of PPE, you think of a mask, a face mask, and gloves. But for our purposes, in a classroom lab, the two main examples are gloves and a lab coat. Now, whenever you're wearing PPE in a laboratory, you have to make sure to follow proper rules. Because, for instance, lab coats and gloves should only be worn in the lab. You should never wear them out in the hallways, to the bathroom, to grab a snack, or anywhere else other than the lab. In fact, whenever you're wearing a lab coat in a lab, you should store it in that laboratory. Okay, so for instance, when we teach microbiology at the college, we have the students put their lab coats and their supplies into big Ziploc bags, and we store them in containers in the lab room itself, because you never want to bring home with you or into your car near other people these kinds of PPE that might have been contaminated in the laboratory. You also have to ask yourself, what should I never touch with gloves or, with the lab coat itself? Usually we're talking about gloves for this one. What should you never touch with gloves? Anything that's not part of your experiment. Specifically, never touch doorknob handles and never touch your personal belongings. Okay, so while you are wearing gloves, it is not okay to touch your cell phone or even to push up your glasses. I know, when you wear glasses, sometimes it's reflexive to kind of push up your glasses or to pull your hair back. Never do any of these things while wearing the gloves, because the gloves quickly get contaminated in experiments. You may not be able to see it, but now they might have some of the reagents you handled, some of the organisms you touched. So you never want to have your gloves touching any part of your body, any of your personal belongings, and anything like doorknob handles, because then you put other people at risk as well. And as soon as you're done with your gloves or any experiment, you always want to toss those gloves in what we call biohazard labeled pails. So there's always an appropriate container in a laboratory that is labeled for you to dispose of gloves. You never put laboratory waste in a regular garbage pail. Okay? Then we have clothing safety restrictions, and a lot of people kind of take this for granted until they end up in a really gross situation, which yes, has happened to some of the students. It's even happened to me before during my time being trained in the sciences. Now, the first rule with clothing safety is that you never want to wear open-toed shoes in a laboratory, and that even includes wearing socks with sandals, which you really shouldn't do anyway. Fashion faux pas. But anyway, when you have open-toe shoes, you are now putting your skin at risk for getting spilled items on or things landing on. And now I always tell the story of how I learned this lesson the hard way: never wear open-toed shoes in a lab. Because back in the day when I was doing my PhD, I was working in a lab, and at that point, when you're a graduate student, you think you know everything. And honestly, by that point, I had worked in laboratory settings so long that I knew that I was very careful, very quick about my experiments, very focused. Now, I was doing an experiment in the summertime where I had to go back in every few hours and check how much the cells had grown. Now, one of the readings, the last reading of the day, was to be taken at 11 p.m. at night. And so it's summertime, you're out and about. I didn't realize how late it had gotten. And I realized, well, I had to get back to the lab really quickly because it was almost 11 p.m. Unfortunately, because it was summertime and I was out and about, I was in sandals, which I normally would never wear to the lab. But I figured, you know what? It's 11 p.m. Who else is going to be there? I'm just going in to put one sample into one machine. Boom, that's it. What can happen? I'm always really careful, so I figured, okay, this shouldn't be a problem. So I go in to do that experiment. And even though I am incredibly careful, or at least think so in my mind, I didn't realize that sometimes the people who do experiments around you may not be as careful as you. And even if they may not be there at that moment, they may have done something that can put you at risk once you're in that lab. So unfortunately, what happened was I go to read the sample and I go to move our usual waste container away from the machine so that I could do my experiment. And what I didn't know is that someone had broken the waste container. There was a big hole at the bottom, but rather than telling someone and cleaning it up properly, they turned it in a way that you could not see that it was broken. So then when I moved this container, it exposed the hole and all of that nasty, gross liquid waste spilled all over my feet. And since I was wearing sandals, it got all over my skin. Thankfully, there was nothing highly acidic in it at that time, but there was a lot of gross stuff in it. So I had to thoroughly do the proper wash down of my feet, and I was grossed out for the longest time. So you may be the most careful person in the world, but you never know about the people around you. So please, whenever you get into a laboratory, even though we won't be going in person this semester, but as people in the sciences, you will in the future go into other laboratories. So please always make sure to wear closed-toed shoes. And in addition to protecting your feet, protect all of your skin. So never wear any kind of clothing that exposes skin. Okay, so no shorts, no dresses, skirts, tank top, any of that. Even though you think, well, I'm wearing a lab coat, so it's fine. No, even with a lab coat on, you do not want to have that underneath your lab coat. The last rule when it comes to clothing safety in a laboratory, very important, if you have long enough hair, tie it back before the experiment, because if you think about it, what does long hair do when you bend toward an experiment? Boom, it flops right in front of you. Now, one time in genetics lab, when the students were working with fruit flies, I noticed that one of my students did not follow the rules. She had her hair, beautiful long hair, down that day and did not put it up in a ponytail like I had told them to do before the experiment started. So in my head, I'm like, well, I could call her out on it right now, reprimand the student, and they'll put their hair up, and okay, that's fine. But what happens? Next time she'll probably do the same thing again, forget as if it never happened. So she needed to learn the hard way, unfortunately. I sat back and I let her see what happens when you wear your hair down. She was working with flies that day where they had to knock them out with the anesthesia and separate the male versus female flies. So what do you think happened when she put her head forward to look to count those flies and separate them? Her hair fell into all of the little fruit flies. And I can guarantee you the next time she was in class, she made sure to put her hair back. In fact, everyone in that classroom always remembered to put their hair back because they saw that happen. And that's a way to learn a lesson where you will never, ever forget that in a laboratory, your hair must be tied back. And in genetics lab, it's only fruit flies. So sure, you might get grossed out, but there's no true danger. But in other laboratories, you work with Bunsen burners, you work with fire, and you work with live dangerous bacteria, like I said, the BSL-2 type of bacteria, you do not want your hair going into fire or bacteria. So always make sure in laboratory settings that hair is tied back. The next point I want to make about lab safety is that when it comes to being in any kind of lab, the three things that you will really, really want to avoid, and I know it's tough to do because we are so all reflexively used to having them with us at all times. In laboratories, never ever have food, drinks, or your cell phones. I know we are addicted to our cell phones, so it's like asking you to cut off your hands, but yes, when you are in a lab, you have to refrain from having your cell phones with you, because when you are in a lab setting, food, drinks, and cell phones are all the biggest items that catch contaminants and then make you sick when you are then outside of the laboratory setting. Because think about it, you have your phone on the bench, there can be aerosol, meaning fine air particles from bacteria you might be working with, even though it's E. coli and it may not be the most dangerous of things, it can cause a problem if you're immunocompromised at the time. Or you can get chemicals and reagents on these items that you then get on your skin, in your mouth, or bring elsewhere, for instance, back home with you, around your loved ones and all. So you have to be very, very careful, because these three items end up getting contaminated very easily and can easily make you sick. Now, like I said, in the genetics lab, you're usually working with little fruit flies, but for a lot of other genetics experiments later in the semester, when you're taking a genetics course or in upper level genetics and molecular courses, you end up working with a variety of bacteria, and they're actually live. You may look at a plate with some, beige yuck on it, or you may look at a tube of liquid that's cloudy, and because you can't see eyes or mouth, you don't realize, you don't think of it as living. But these tubes, these plates, they do have living bacteria in them in laboratories and in classrooms. So you have to be very careful anytime you're handling living bacteria in experiments. You never want to shake any of the liquids that have bacteria, because that can create aerosols, like I mentioned before, those fine droplets or particles in the air that you can then breathe in. And most importantly, when you're dealing with plates, bacteria with plates on them, like you would do in a transformation or conjugation, which we'll talk about later in the semester, you always want to open them in the clamshell manner that you see here. Notice that this scientist, instead of completely removing the cover and placing it elsewhere, they open it up like a clamshell to reduce the amount of contamination going on that plate and to reduce the amount of bacteria that you would also be breathing in. So always clamshell any bacteria plates, circle, star, highlight that point that you would never open up a bacteria plate fully. Okay, you would never separate the cover from the plate completely. And always just be very aware of the tubes, of the equipment that you're using. Don't let sterile items touch the benches or your body. Just be very, very careful to keep everything as organized and sterile as possible. Also, whenever you're carrying liquid tubes like you see in this picture, you always want to carry them on a rack, just like they are in the picture. Now, thankfully, if you do spill in any of your laboratories, you can always use disinfectant to help clean it up. What you should always do in that case is you immediately spray the disinfectant on the spill and leave it for about 15 to 20 seconds. Even if you're not impatient, you could go a little bit longer than that too, just to really make sure that it's doing its job of killing those bacteria, getting all the little crevices. And then wipe up the mess. And what's the last thing you should do? Make sure to change your gloves. Throw out those gloves. They probably got contaminated, and take a new pair anytime you have spilled anything. Okay? And always let your professor or coworkers, if you're working in the lab, know if a spill occurred, just to make sure that everything is done proper and that no one ends up hurt or sick. Now, the last point I want to make with lab safety is that you should always be spraying down your benches even when there's not a spill. So you should spray down your benches before you start an experiment and after, because again, you never know how careful other people are. So it's best to be as careful as possible for yourself. So spray down before working there to make sure to clean up anything anyone else may have kind of left there, and then spray at the end. And always, always, always wash your hands at the end of any laboratory experiment, even though you were wearing gloves, because proper hand washing hygiene is the best way. It is the most efficient way to break the chain of infection. Okay, so always be good about washing your hands. Okay, so now we get to the fun stuff, the introduction to our genetics lab. And when we're working in the genetics lab, the first thing you want to ask yourself is, well, what model organism will we use? In this case, which model organism will we use to study heredity? Now, whenever we mention a model organism, basically that's an organism that you'll use in experiments to help you better understand concepts that you can then apply to humans or other organisms as well. Now, the one that we will use, I wrote it out so that you know how to spell it in your notes, is Drosophila melanogaster. And that's basically a fancy way of saying fruit flies. So we'll usually just call them either Drosophila or fruit flies. Now, you may ask yourself, well, why are we going to use fruit flies in our experiments? I usually just squish them when they're coming too close to my bananas or apples. Well, the reason that we use them in our lab classroom at a university is, think about it. First of all, you want to write down that they have a short, speedy, or quick lifespan. Usually within 10 to 14 days, you're able to get completely new adults, okay, which is great in a classroom where you only have 13 to 14 weeks to do your experiments. They're also cheap, meaning inexpensive. You want to circle star highlight that one, because in universities or in classrooms, you want cheap because you have so many students, so many sections of classes. The place would go bankrupt if they were expensive. It's also great that they're small in size. Think about fruit flies when you've seen them, right? They're big enough for you to see with your naked eyes, meaning no need for a microscope. But they're also small enough that in a classroom, you could have one little vial and that little tube will have like 100 of them, which is great for sample size. You want big sample sizes for experiments, okay? So they have a quick lifespan, they're cheap, they're small, and they have minimal needs in terms of food and shelter. Basically, we put them in a little tube with some sugary media at the bottom, basically sugary food at the bottom, and they're good to go. Okay, they can stay in that little tube with their food and they're fine. They're also excellent because they have obvious phenotypic differences, which we'll talk about in later slides. It basically means that genetics-wise, you can clearly see the color of their eyes, the type of wings they have, the type of bodies that they have. So they're great for experiments. Okay, so I'm going to give you a recap slide where I wrote down all of those things just in case you missed any of them. Okay, so here is a recap slide of the notes, basically like closed captioning, transcript of what I just said. I also threw in at the bottom that it's kind of good that fruit flies are not really toxic. Sure, you know, you might inhale a little too deeply and kind of maybe one day I'll accidentally have a student choke to death on a fruit fly or something, but usually they're not really causing problems in the classroom. They're not, you know, as toxic as or as problematic as some of the other organisms you might work with, like bacteria. Now, in the previous slide, I mentioned that the fruit flies have a pretty quick, speedy life cycle, and that's what part of why they're so good for us to use. Now, here I ask you how long and exactly what the stages are. So the first stage that you're going to see is the egg stage, which you can see right here what the eggs look like, little white kind of oval shaped pellets almost. And that egg stage will basically be about 1 day, okay? Then they move on to the larva stage, and that's the term I always want you to use for these guys. Now, some uncultured people may call those little guys maggots. I never want to hear you use that term because I find it offensive to my babies, the fruit flies. Okay, so they are larvae. And the Drosophila will basically stay in the larva stage about a week. Now, keep in mind these times that I give you, 1 day, 7 days, it also depends on factors like the temperature in the room. If it's a really hot room, then the organism is going to go through its cycle a bit quicker. So it might not need a whole 7 days in order to move on to the next stage. In some cases, it'll be quicker in the heat. And the opposite is true if the room is really cold. In cold, metabolism reactions slow down. That's why if you've ever gotten a fly stuck in your fridge, you open up the door and you notice that it's very slowly moving to get out. Whereas if it's ever been a hot summer day and you see a fly inside your house, it's bouncing off the walls. It's going ultra fast because heat speeds up reactions, cold slows them down. Okay, so now getting back to this cycle, you have the larva for about 7 days, and that's what you might commonly think of as maggots. They're moving around a lot. And then you get to what's called the pupa stage, and you see that here. Okay, looks like a little cocoon, it's a bigger pellet than the eggs. And a lot of times when you culture fruit flies in vials in a laboratory, these you'll see stuck to the sides of the wall or to any netting or nets that you put inside there. Okay, so they're immobile, not moving pellets basically. They look like little cocoons. That's the pupal stage. And the pupa stage will usually only be about a couple of days. So that's about 2 days. And then you end up with the adult fly, okay, full-grown fly, okay, and that's the one that takes on a new wonderful trait. It has wings, so it can fly, right? Hence the name fly. It is a little fly. Okay, now once it's at that adult stage, it tends to live about 20 to 30 days unless you squish it. Okay, just kidding, unless it encounters any kind of, environmental stress. So we'll put about 20 to 30 days. Again, all of these days that we list out, they're not exact, meaning for some flies, they may be quicker, may be longer, it depends on the fly. Now I then ask, so we have the length of each stage. We have the stages themselves. So notice there are 4 stages. We had egg, larva, pupa, and adult fly. And then I ask you how you can tell the difference between certain stages. And basically what I mean by that is that the egg stage and the pupa stage, these, like I said before, are what we call, the pen slipped, immobile. Okay, immobile, meaning not moving. Okay, they will not be able to move. You won't see them slinking around or moving. Whereas the larva and the fly, these are mobile. Okay, moving. They move around. They move around and they gross you out by doing so. Okay, so both of those stages will move, whereas the other stages will not. And like I've said, if at any point you have any questions, you're unsure of any of the topics we discussed on this slide, just send me a message in the Remind app. Now, a lot of times in the genetics lab, when we're working with fruit flies, what we're actually doing is mating them together, which basically means that you need to be able to distinguish between male and female flies and separate them out so that you can properly organize your experiment. Now, thankfully, fruit flies have some, traits or characteristics that make them pretty easy to tell right away whether they're male or female. One of the first things we can notice is that the males are smaller, whereas the females are larger. So if you look at this picture over here, you'll notice that both of these are actually adult flies, but this one here is smaller because it is male. Okay. Now, a trick to remembering which is which, I always think of, well, the female is the one that gets pregnant. So think of pregnant as being much bigger, carrying all those babies, right? So females are larger. Another difference that you'll notice in this picture is that the males have what we call a round, blunt rear, whereas the females will have more of a sharp, protruding rear. Rear being a fancy scientific way of saying butt. Okay, so you'll notice the shape of this male's back area. Sorry, my pen is dying. The back is rounded, whereas the female's is more sharp and protruding. Now, also, while looking at those rears or butt areas, you'll notice, look at this colored in area, right? That's much darker, whereas this area is basically white. You don't have that black patch. So for males, you notice a black patch on their rear, whereas the females have a white patch, okay, on their rear. Okay, so black patch versus white patch or no color on the female. Okay, now the last thing is the most definitive and one of the main things that we use, because if you think about it, well, who's to say whether a fly is small or large? You know, if you have another fly next to it, sure, you can kind of figure out a little better. But if it's just chilling by itself, it's kind of tough to tell that, okay? And in terms of the rear area, well, it's easy when that fly is flipped over the way they are in the picture on this slide. But when you're just looking at it standing in front of you, it's a little tough to tell. So the real main definitive difference that we use is called sex combs. Okay, and so that is sex combs, okay, specifically on what we call their forearms, meaning their front legs. And females have no sex combs. Now what that means is that when you look at a picture of a fruit fly, you look at their front legs, and it's a little tough to see on this bottom picture here, but this fly's front legs, if you zoom in on them, has a black spot on each, okay? So when you look at the front legs of a fly, if you see that black spot, which when we zoom in looks like little combs stuck to them, that's a male fly. So whenever you look at a picture of a fruit fly, the first thing I want you to look at the front two legs. Notice how the female in this picture, the legs are just completely beige like the rest of her body, whereas the males will have these little black dots, okay? So first thing to look for, if you see those on a fly, then you would say that fly has sex combs on its front legs, so it is a male, and that's very definitive. You can't kind of go wrong with that one. Whereas small versus large, round versus sharp, sometimes those are tougher to tell. Okay, if you have any uncertainties, any questions, just let me know in the Remind app. Okay, I want you to stop everything, right? Stop everything, grab your phone, head to the Remind app, and I want you to answer the question I'm going to say right now. I want you to message me in the Remind app or on the Remind website and tell me, look at this fly right here. Tell me whether you would categorize it as male or female and explain exactly why you would categorize it that way, okay? So pause the video, make sure to send me the message right now so that you do not forget. Again, simply message me, is it male or female, and why do you say that? What makes you categorize it one way or the other? Okay, so moving on again toward the heart of genetics. One of the terms you'll encounter a lot in genetics is the word phenotype. And what I want you to do is circle or underline the PH in phenotype, and then above that write PH, circled or underlined, in the word physical. Okay, so write physical characteristics and circle or underline the PH in physical as well as in phenotypes to remind yourself that phenotypes are what you physically see. So for instance, it's the expression of genes or the genetics. So for instance, do you see someone having blue eyes or brown eyes or green eyes? Those would be their phenotype for eye color. In the case of insects, do they have long wings? Do they have short wings? Do they have no wings at all? Those are all different phenotypes. They're physical expression of the genes. It's what you see for their genetics. Now, when we talk about the fruit flies, there are two types of phenotypes that I want you to be aware of. Okay, there's the term wild type, and then there are mutants. A wild type fruit fly will have three characteristics that I want you to know. A wild type will have large red eyes, so you can see that here. This is a wild type fruit fly, large red eyes. It will have a normal yellowish brown body that has little black stripes on it. So notice that it's a yellowish brown body with black stripes going across, okay? As opposed to this guy over here, one of the mutants, notice there are no black stripes, okay? So wild type has the big red eyes, the normal yellowish brown body with black stripes, and it will have oval wings that are longer than its body. Notice the end of its body is over here, and the wings are longer than the butt of that fly. Okay, so wild type flies, I want you to circle wild type and write those three traits: large red eyes, yellowish brown body with black stripes, and oval wings that are slightly longer than the body. Then for mutants, circle mutants and write that means one or more traits vary from the wild type. So either the eyes will be different, or the body will be different, or the wings will be different, okay? One or more traits differ from the body type. Now, the way that fruit flies are named when you have these wild type and mutants, they're actually named for their abnormal trait, meaning they're named after whatever trait looks different from the wild type. So for instance, here you have a wild type fly. This fly next to it, what do you notice is different? The eye color, right? So what would you name this fly? It's a white-eyed fly. because it's named after the trait that's different from wild type. So this is a white-eyed fly. This is a sepia-eyed fly. This is an apterous fly. They're my favorite. Look at that little guy. Notice, what is it? It has no wings. They're great when you work with them in the lab because they can't get away from you. Okay? I meant that in a non-stalker sounding way. So they have no wings, they can't get away, and they're very easily identified as a mutant. Okay? And they're named apterous for having no wings. The last one here, if a mutant has multiple traits that differ from the wild type, it'll have a multi-part name. So this fly here that we use in what's called a 3-point cross, this is called a yellow-white miniature fly. Yellow because the body, unlike the wild type, doesn't have those black stripes you see in all the other flies. Instead, it's just yellow, the body. Yellow-white because, what do you notice? White eyes instead of the wild type red eyes. And miniature because the wings are miniature, meaning these wings line up with the butt of that fly. They do not extend past the body of the fly the way the long wild type wings would. So this is yellow, white, miniature. Okay, yellow for the body, white for the eyes, and mini for the wings, all the different mutations it has. Now in class, what we use these different mutants for, the apterous and the sepia flies. So apterous is no wings, sepia is dark eyes. These flies we usually use for the monohybrid and the dihybrid cross, which we'll talk about in upcoming weeks. They represent recessive traits, which means traits that can be masked by the dominant trait. Okay, so a parent can have the allele or the genetics for a sepia-eyed baby and yet not look sepia itself because the recessive trait can hide. Okay, so they're recessive compared to the dominant wild type. Okay, wild type is dominant. The white-eyed fly, we use this one in X-linked, or we also call them sex-linked crosses, meaning you'll find that trait on the X chromosome. And the last one we mentioned here, we use this in the 3-point cross, which again, you'll understand these terms a little more in the future. Now I'm going to give you a recap slide because I have said a whole lot on this one slide. Okay, so here's a recap slide. Since I said so much on the previous slide, you can pause, jot down your notes, and then continue on. Now, the last thing I want to talk about is a little bit about Mendelian genetics, or as you've heard, Gregor Mendel initiated genetics. Now, like I said before, two terms you'll encounter a lot in genetics are genotype and phenotype. And the difference is that genotype is the actual genes, okay? The genes or the alleles for a particular trait. So let's say we were talking about sepia mutant flies. Well, we take the letter of the mutant, so sepia is an S, and we would say a genotype might look like SS, two lowercase s letters. The reason why this genotype that I'm writing for that trait is 2 letters is you inherit one allele from mom, one allele from dad. So we write SS, meaning that this fly inherited a mutant allele from mom and a mutant sepia allele from dad. And those are their genes, their genotype. Phenotype, on the other hand, what did we say? When you see PH, think physical, right? So the physical trait, okay? It is what you see. So in the case of a sepia fly, the genotype would be SS, but the phenotype would be sepia, okay, those dark eyes. Okay, now, like I just said, you heard me use the term alleles, okay, the term alleles, okay. What that means is basically each trait has variations. So sepia versus wild type red, those are two different alleles. The classic example with plants, plant experiments, is tall versus short. Okay, so alleles are simply variations of a trait. Okay, so for instance, tall versus short, or sepia versus wild type. Okay, so make sure you're comfortable knowing the terms genotype, phenotype, and alleles. Okay, circle, star, highlight that word as well. Each of these is an allele. So 1S, 2S, each of those is an allele. Together they make the genotype. Okay, the genetics that we refer to that organism by. Now, the inheritance of these alleles, okay, these individual variations of a gene, the inheritance of these alleles is governed by what's called Mendelian laws. And there are two laws I want you to be clear about what they are. There's the law of segregation and the law of independent assortment. And both talk about how you're inheriting alleles from your parents, but they have two very different meanings. So we're going to draw them out on the next slide so that they're a little clearer. Again, if any of this is confusing or I went a little too fast, always feel free to contact me and remind, and I'll go over it again. Okay, so like I said, there are those two laws of inheritance, both dealing with alleles, meaning the variations of a gene, you know, the genes that you inherit from your parents. One of the big differences between the two, I'm going to underline S in segregation to remind you that this is a law describing the inheritance of a single trait. Okay, so for instance, with the fruit flies, maybe we're just looking at eye color. Whereas the law of independent assortment, that one's looking at the inheritance of multiple traits. So for instance, eye color and wings. Okay, so now first looking at the law of segregation. What it means is when you're looking at a single trait, for instance, eye color, okay, we're looking at those sepia colored flies, for instance. What this law says is that alleles will separate from each other during meiosis. Okay, so alleles separate from each other during meiosis. And meiosis is when an organism is making sex cells. Right? They're making their gametes. Okay, your gametes is your sex cells, such as sperm or an egg. Now, basically what this means is when you are an organism, you have two alleles for a single trait. You have one allele that you inherited from mom and one allele that you inherited from dad. Okay? Then what happens is you go to make your gametes, either sperm or an egg. So these alleles have to then separate into two different gametes. Okay, so this one will go to gamete 1, and this one may go to gamete 2. Now the reason your alleles have to separate, think about it. If you are making a sperm cell or an egg cell, what's that sperm cell or egg cell going to do? It's going to fertilize. It's going to meet up with another gamete, right? So for instance, if you're making a sperm cell, it's now going to go find an egg cell and fuse together to make a baby, right? Well, if your alleles, your genes in your body did not separate so that you are putting half of your genetics into that single sperm cell, then what would happen if you put all of your genetics into one sperm cell? When it met with an egg that has all the genetics, the organism created would no longer be a human. It would have twice as many genes than it needs. So instead, you want only half of your genes going into your sperm cell so that those half will fuse with the half of genes present in an egg, and now you get one whole human. Okay, so very important. The law of segregation is very important, that for each single trait, one allele is going to one gamete, the other allele is going to the other gamete. Now, the second law of independent assortment, that's for multiple traits. And what this law says is genes on separate chromosomes or at great distance on the same chromosome will assort separately. So first I'm going to write that down for you. So genes on separate chromosomes or at great distances, on the same chromosome will assort separately. Okay, so what that means is, let's say an organism, so sometimes, let's see, we did SS, we've already looked at eyes, so we can have one trait for eyes, right? One trait has two alleles, one from mom, one from dad, and then we're going to look at, let's say, wings. Okay, so we'll put two alleles for wings. A, because we're thinking apterous. Either the fly will have wings or it will be apterous and not have them. So what this means when you have this one organism, so this is one organism, two traits, and each trait has two alleles. What this means is that our capital letters do not have to be in the same gamete. Instead, when a sex cell or gamete is being made, all that matters is it's getting one allele from each trait. So it gets one allele, either capital S or lowercase s from eyes, and it needs one allele, either capital A or lowercase a for wings. Okay, because you want the gametes to have one allele per trait. Otherwise, what happens? You are going to get someone with extra genes in their body. They're not going to be human. So you only want one allele. So when this organism, this one organism, so organism, okay, that's one organism with two traits. When it goes to make its gametes, it can make four different gametes. It can make a gamete with a capital S for eyes and a capital A for wings, or it can make a gamete with a capital S for eyes and a lowercase a for wings. Or what else can it make? It could get the lowercase s, right? Because all it needs is one allele from each. So it could have a lowercase s for eyes and a capital A for wings, or lowercase s for eyes and a lowercase a for wings. Sorry. Because the allele pairs separate independently. Okay, so for possible gametes. Sorry, my pen is dying again. 4 possible gametes, okay? Because the allele pairs separate independently. Meaning that capital does not have to stay with capital, lowercase does not have to stay with lowercase. All you need to know, all you need to have is one allele for eyes, one allele for wings. It can be either one that you end up getting, okay? So basically, the traits will be transmitted independently of each other, and you'll get to really see this when we talk about the dihybrid cross, okay? So again, if either of these laws, what they mean, any of the terms that I use, such as meiosis, gamete, allele, anything, if anything's unclear, make sure to send me a message in the Remind app. Okay, that's it for today's lesson. Normally, I would have at the end of a lab a few slides with results, pictures or result numbers for you to then analyze in your post lab. But for this week, because it's just an introduction week, there are no results for you to analyze. Instead, it's a regular old post lab assignment for you to start really thinking about what we went over in this lesson and to really see if it made sense to you. Now, please keep in mind during post lab assignments, which you'll find in the same folder as this lesson video, the post-lab assignments are basically what you would be writing down or doing in the classroom at the same time you had the lesson in front of you. So you are allowed to use your notes and the narrated video as you do your post-lab, okay? But do not use your cell phone, your internet sources to do so, okay? If I find internet words or if you copy from one of your friends doing a post lab, that will count as cheating and you will automatically fail. They're very easy, the post lab assignments. All of the answers are basically in the video. Now please keep in mind that quiz number 1 will be next week. You will be able to access it between Monday 8 a.m. through Wednesday 1 p.m. So anytime during that time frame, you are allowed to click on the quiz, take the quiz, but after Wednesday 1 p.m. it will completely disappear. Okay, so you will no longer have access if you do not take it by Wednesday 1 p.m. You have now missed that quiz. It becomes a 0 and there are no makeups. But because I know that sometimes life happens, it's very understandable. You may not be able to make every quiz. I do drop a quiz at the end of the semester. Okay, so if you miss a quiz or you do poorly on one of the quizzes, it will be dropped at the end of the semester. Okay, but make sure you notice it's only one quiz that happens with, okay? The quiz format will be 20 multiple choice questions. There will be a timer set, so as soon as you open the quiz, the timer will start counting down. If you leave Blackboard, you leave the quiz or anything, the timer will still continue and will automatically submit once the 25 minutes is done. So please make sure to do the quiz in a timely fashion. Once you open it, you have to complete it, okay? Otherwise, it will just submit and you will have missed answering some of the questions. Please keep in mind that there is no backtracking. So do not leave any question without an answer. Make sure to pick an answer and then move on to the next question, as you will not be able to go back. The quiz number 1 will cover any material covered in this narrated lesson. Okay, so anything is fair game, whether it is lab safety information, whether it's stuff we talked about in the intro to genetics or the Mendelian genetics. Again, if you have any questions, anything at all, please contact me in the Remind app anytime. Like I always say, I have no life. Okay, have a wonderful day. Thank you.